My name is Tyler Huth, and this is Structure-Function Study of Methylmalonyl-CoA Epimerase. Our lab was interested in looking at exactly where do our medicines come from. In Figure 1, a pie chart visualizes the major sources of compounds that become targets for medicinal products. Although in today's pharmaceuticals, synthetic products have become the more common route to drug development, natural products like those found in bacteria and plants make up nearly a third of the source material. We are interested in a biosynthetic complex called a polyketide synthase, or PKS, which produces the natural product antibiotic erythromycin. Often fermentation of natural products, like erythromycin, can be time-consuming and require further synthetic derivation. But what if we could harness nature's machinery to circumnavigate the issues with natural fermentation and express these natural product derivatives in vitro? This project focused on one major component of the PKS mechanism that involves the enzyme methylmalonyl-CoA epimerase, or MMCE. PKS is stereoselective, meaning it will only accept one conformation of its natural substrate, methylmalonyl-CoA, in a 2S conformation. Figure 2 displays the key MMCE reaction which interconverts between 2R and 2S to successfully incorporate the necessary substrates into PKS to complete its biosynthesis reaction. In order to accomplish an accurate understanding of how stereospecific substrates are prepared for PKS, we aim to study both structure and active site chemistry of MMCE. To accomplish this goal, first we expressed and purified MMCE from E. coli. Following purification, we generated a tool set of transition state analogs that mimic MMCE's natural substrate and essentially freeze the protein in the intermediate position. MMCE was co-crystallized with the natural substrate, the transition state analog that contained a nitro group, and no substrate at all. Post-crystallization, the crystals were looped and flash frozen in liquid nitrogen to preserve them until arrival at Argonne National Labs in Chicago. At Argonne, an X-ray beamline was shot through the crystal samples and a diffraction pattern was generated. These diffraction patterns can be solved and refined to obtain electron density maps that provide accurate atomic structure information at high resolutions. Figure 3 shows an overlay of these electron density maps of the empty or apo active site, the natural substrate bound to the active site, and the nitro analog bound. Several residues are labeled in the figure, and these residues have been identified to be close enough to be catalytically relevant for any MMCE active site chemistry. All the atomic structures were solved at a high sub-2 angstrom resolution and provide useful insight into targets for mutagenesis. Mutagenesis is essentially where we would remove a residue altogether to understand what that residue contributes to enzyme activity. Now, Figure 4 shows a previously proposed or older mechanism for MMCE by another lab, and they hypothesized based on activity data but no supporting structural data like what we have here. Their hypothesis proposed an acid-base mechanism with protonation done by the glutamate residues, GLU 134 and 43. But our data shows that the glutamate residue, indicated by the red arrow in Figure 3, moves farther away from the active site in the intermediate position and supports an alternative hypothesis to that was proposed as an acid-base catalysis. The second portion of Figure 4 displays an alternative mechanism proposed by our lab in which MMCE undergoes an enolization and subsequent tautomerization. Activity assays can help us determine the validity of this alternative hypothesis and further develop an understanding of what kind of chemistry is being used at MMCE's active site. Following the determination of MMCE structural data, we seek to understand the active site chemistry of MMCE more thoroughly. In order to know which active site amino acids are directly responsible for MMCE activity, we will generate a set of MMCE mutants in which a single amino acid will be changed to a neutral amino acid. Following mutant generation, an activity assay will be created and optimized in order to measure corresponding catalytic rates and help generate a feasible hypothesis on the MMCE active site chemistry. The elucidation of MMCE and the neighboring PKS's components, structures, and functions will guide future bioengineering projects in their efforts to commercially synthesize natural product derivatives and open a new route to future drug development opportunities. Thank you.